Morning. So welcome also to the University of Illinois for those who are not from here. The picture you see here is the Neuropsychiatric Institute here, which after Columbia's is the second oldest in the country, except ours is shaped like a brain. And is this a pointer here? Oh, this is different. Okay. So there's what we call the North Tower, which is the left hemisphere, has neurology and neurosurgery together. We combine this with psychiatry in the South Tower. And you guys know what the connector between the two hemispheres is called, the corpus callosum, right? So I want to tell you today about what we've been doing for some time in human epilepsy. And many of you know people or dogs or pets or seizures is a very common disorder, about 1% of the world's population. And really, it's a disease that can occur after many different brain insults. Just about anything developmentally or trauma or stroke or anything that can happen to the brain can lead to epilepsy. And it's a disease of connectivity where thousands, if not millions of neurons start firing synchronously in ways that they should not, creating abnormal discharges or field potentials. To this day, we can only measure these by putting electrodes either directly on the scalp or directly on the brain surface to measure these millions of neurons that are firing within milliseconds, producing this disease. When these epileptic discharges become synchronized and repetitive from, say, a focal area, as you can see here listed by a focus, they spread through the brain and produce a convulsion, a shaking seizure that you think of clinically. A lot of seizures are actually quite subclinical as well. So when I got into this business, surgery was really the only way and still remains the only way to cure epilepsy. If you can identify that focus and safely remove that focus, you can cure epilepsy. Drugs we have right now, they can suppress seizures. You miss your pill, though, you have a seizure. So as part of these fairly extensive surgeries, we don't want to take out the wrong parts of the brain. You want to take out the abnormal parts. So we do extensive intracranial mapping. The signal that we see on the surface of the scalp is really not strong enough to localize these foci. So the skull comes off, hundreds of electrodes go on the portion of the brain that we think are close to where the seizures are starting, as shown in this picture. So each of these little steel discs there is an electrode recording. There's wires coming out, and you can actually record in real time. The patient goes back to the hospital room, and we record for about a week of continuous data showing areas that produce seizures. You can see some of these channels here. And you can see other areas that don't have seizures. So tissue is removed to hopefully enact a cure for that patient. Now, one of the major challenges is that there's like TMI, too much information. The amount of information we get from these long-term recordings, we really don't know what it is and what to do with it. And you see all these little squiggles like this. The little green lines you can see, those are 1-second intervals. And you imagine going on for a week of recording, that's a ton of data. So that's big data in itself. So we spend time actually trying to take this noise and turn it into actual quantifiable measures. We have programs that can mark what we call the spikes. That's the synchrony of the neurons firing. We take those locations and the frequency of those spikes, and we plot it back on the three-dimensional brain surface. Now, everybody's brain in this room looks different. You don't have the same shapes and things, so we have to take that individual's brain, build a 3D model of it, superimpose the locations of the electrodes with these little spheres on the surface, and then we can pseudo-color areas that have a high frequency of these epileptic spikes that happen between seizures in red and areas that don't have high spiking in green. And you can actually now start to see that the spatial organization of this epileptic activity sometimes actually obeys some of the spatial patterns of the brain structure. So today, this symposium is on personalized medicine, and I'm going to talk about this as a very personalized approach because we get so much information on a single brain, on a single patient. So we have these electrically mapped human brains, and the amazing thing about this procedure is we remove tissue to cure the disease. And that gives us an amazing opportunity to actually examine that tissue. If we can link all these variables together, we have an enormous opportunity to understand the disease, to understand the brain, with so many different attributes put together. So what I'm going to talk about today is how we've taken advantage of this amazingly unique opportunity to understand the human brain on a personalized level, and then to use this to find what we call common mechanisms or common pathways, because developed treatments, it's great if you can do an individualized CRISPR and everybody has their own mutation, but it would be much nicer to have something that works for everyone. So starting with personalized and using this to find common pathways. So how do you do this? Well, it takes a lot of work and a lot of meticulousness. So when, say, a patient has surgery and say they have this temporal lobe here removed as part of their surgery, these areas of tissue, we have to know exactly this piece of tissue that was, say, the high spiking area. We have to identify that precisely. Once we get the tissue, we want to be able to subdivide this in a way so we can get the most information from it. So the first thing we do is we cut it in half. We take half of it, we fix in a way that allows us to do in situ hybridization, immunochemistry. The other half we freeze. We then take even segments of the gray matter from layers 1 through 6, alternate slices, to look at protein, RNA, and metabolites. And this way you can then link these molecular variables to the histology, to the brain location, to the brain imaging, to the brain physiology, to the clinical health record. And that's really the sort of systems biology project that we've kind of put together. So the type of information that we collect at the organ level would be looking at where this was, what was the imaging characteristic, what was the physiology, what are the quantitative measures of what was going on at that precise location, what did the histology look like, was there a developmental abnormality, was there a lesion, what's going on at the genetic, we heard about genes and DNA, are there epigenetic changes in the DNA, are there changes in messenger RNA levels, changes in protein levels, modifications of proteins and metabolites. Wouldn't it be great to have all that information on these cases and then somehow put this together? One of the challenges, how do you put so many different types of data together? I was talking with Dr. Golden, my old partner in crime for many years. There's wonderful databases on each of these individual things, health records, pathology servers, but we need to bring these together to create value. And so that's basically what we did. And this is an outline of our database structure, from clinical research to the physiology to the imaging, inventorying tissue that comes out, and then any of our molecular, what we call omics, on those tissues. And the goal, of course, is putting all of this information together in a way that you can mine it to identify biomarkers, drug targets, and develop diagnostics and therapeutics. So let me talk through how we've kind of done this. So this is a sample web page from our server. This would be a clinical page where we say that this is a 33-year-old male with epilepsy. These are the medications the patient was taking. These are the finding on neuroimaging. I know it's probably small, so you can't read it. This is the outcome from surgery. Did we do something right or wrong in surgery? What did we see as terms of the seizures? We do complete neuropsychology studies before and after surgery. So if you want to link what we see on the electrical signals or pathological to behavior, to depression, to IQ, all of this we can do through these data sets. But the key is the electrical information. We don't collect tissue unless we have an electrode over it for at least several days, and we can say whether it was firing, not firing, in that period prior to tissue collection. So this is an example of 10 electrode names per page. We have 9 pages on this particular patient, so over 90 electrodes of information, and these are the quantitative measures of the spiking. We plot that again on that 3-dimensional brain surface so that you can click on a certain area that shows a hotspot. It'll tell you what electrode that is, and it'll link back to the XYZ coordinates to go back to your imaging studies and tell you exactly what the spike or seizure characteristics were at that site. We inventory the tissue, what we have, what we used it for, is it fixed, is it frozen? We keep what we call our 2D images. You need pictures of the brain with and without electrodes for quality control, but we do histological sections and stains and digitized images of every block of tissue that we do, so we can click on it and on a thumbnail, zoom in on that area and actually mine it, or go back to that box of slides in the freezer and do more studies. RNA we make, subcellular fractions of proteins, and we'll look at maybe 43,000 genes on a microarray and put that in the database. So in this case, we have 10 genes per page, 4,350 pages. Do the genes go up? Do they go down? How do they compare? And what's the statistics? So this is our version of big data. It's not thousands and thousands of patients looking at a single area of the chromosome and what a gene is doing. It's looking at an individual and looking at thousands and thousands of genes, different brain locations, different electrical signals, but creating a format to tie these pieces of information together. And the major challenge, as we know with all big data, is this: finding the needle in the haystack, finding something useful from this information. And it's how you put it together and the quality of data that determines whether you can find the needles. So now I'm going to give you some examples of some of the needles that we've been finding. So here's a simple question, something we don't know the answer to. Here's a part of the brain, we say seizure onset. That's the part, that's the focus that causes all the trouble that starts the seizures. Now often with these surgeries, particularly with some of the children, very large resections are done. And we also have tissue that doesn't have seizures on it, that doesn't have a seizure onset. So you have to ask, when you look at something with 40,000 variables, what's the best control? It's not going to be another patient who died in a car accident and you have some random piece of brain that was post-mortem rather than fresh. There's too many variables that are different. So you find a piece of tissue, if you can, from that same patient that has less disease or no disease on it. And if it's removed as part of the surgery, you have an incredible opportunity to ask what's the difference between those two areas of the same human brain. Why does one area produce seizures and another area not produce seizures? So this is a relatively old technology, but it's actually still a very, very precise technology to look at gene expression changes called microarrays, where each spot on this is a different gene covering the whole part of the coding human genome. If a spot turns red, the gene goes up. If the spot turns green, it goes down. If it's yellow, it's unchanged. So very rapidly, you can do this in replicate and get some very tight statistics on which genes are changed between areas that produce seizures and areas that don't have seizures. So this is what I call the personalized transcriptome. So for this patient, say the green circle here, you may find a group of what we call differentially expressed genes between the area that produces seizures and the nearby area that doesn't. And you can say, well, that's interesting. What do we do with it? Well, you look at the next patient and say, well, if we compare these two transcriptomes, is there any overlap? And again, turning this personalized transcriptome into a common pathophysiological set of genes that may indicate why one area of the brain produces seizures and another doesn't is what we do. And you can do this with as few as 5 patients and get really, really, if you see anything left in that intersection, it's not by chance and highly validated. So from this study, from 43,000 genes we looked at, we simplified our problem of why areas produce seizures to 137 genes, a much more tangible number of genes to be able to mine and try to understand. We do things called ontology, which you probably know about, to find what pathways are known, what are those genes known to do functionally, and statistically, is this happening by chance, or is this a unique pathway? So by building these models from epileptic activity, we have certain pathways such as MAP kinase signaling, certain transcription factors like CREB, and certain pathways like plasticity pathways in immediate early genes that just pop up overwhelmingly. We call this our low-hanging fruit. So it doesn't say what causes, what affects it, but these pathways are highly statistically associated with areas that produce seizures in the brain. Then we can go back and try to understand why. Also important when you do high-throughput studies is validation, validation, validation. Is it really true? The goal, of course, is to turn these into useful clinical biomarkers, drug targets. We have to do this to get to clinical trials, but to get there, we have to go through a series of steps to convince ourselves and everybody else that the pathways discovered in this way are actually not only clearly related to the disease, but potentially, if you modify those pathways, can actually change or treat or cure the disease. So the first thing we do is we call human validation studies, because we have not only RNA from each brain regions, we have protein samples. If you predict a certain pathway to be activated in those areas, there's ways of looking at constellations of genes. And we heard in the last talk about the importance of editing single genes in some of these rare disorders, but most of our common disorders are not single genes. They're hundreds, if not thousands of genes and pathways. So the more we can kind of take populations of genes and put them together in different ways, I think the closer we are to deal with some of these more common polygenic abnormalities. So particularly since something like MAP kinase and CREB, there's actually very good markers to see if these pathways are activated, the proteins become phosphorylated. So very quickly, you pull out the exact same sample where you predict these pathways to be enriched. You do a Western blot for either phosphorylated ERK in the MAP kinase pathway or phosphorylated CREB, and you see very quickly the epileptic areas have enormously higher amounts of activation, even in this sort of between seizure state than non-epileptic areas. And here you can see the phospho-CREB is active relative to the non-phosphorylated CREB, and you see that's not present in the control tissue. And you just do this on a bunch more samples, and you can convince yourself yes or no a given pathway is actually activated. So we can actually find from as few as 5 patients common pathways, but you start with many, many variables and hone in on them, including the MAP kinase and CREB activation. Another thing we can start to do is ask, okay, so we focused on areas that produce seizures. I showed you these pictures before that most of the time the epileptic brain is not seizing, but you have these abnormal spikes that are going on all the time, shown by these little green areas with little lines under them. Those are spikes. They're happening all over, and we don't know, are those the same as seizures? Are they different than seizures? Do they interrelate somehow with seizures? So we would then take another experiment where we take areas of low spiking tissue and compare to high spiking tissue. Doing that experiment, instead of getting 137 genes, we ended up getting 1,700 genes. The only reason why is because we used 15 patients in our series instead of 5, and with the same statistical cutoffs, you get more genes. So the number of genes and the statistical cutoffs are really arbitrary. The more significant are up here, the lower there. You start at the top and you work down. But what's interesting when you start separating two electrical patterns in the brain, seizures, which are the rhythmic spikes, and individual spikes, you see that there's overlap in these transcriptomes. So that there are some genes that are only present in spiking, some only in seizures, and many combined by both. So if you wanted to make a drug against spiking, you might, and not hit seizures, you could focus here. If you wanted to focus on seizures and not spiking, or hit them both, you look at the intersection of these things. So what now can we do with this? So we have these markers. We can validate these pathways are truly activated. We can actually start to go back to the brain tissue and try to understand. The brain is a very complex structure. We have in the cortex 6 different layers. Where is this happening in the brain? Can we use these as what we call tissue biomarkers to understand a little bit about the abnormal networks that occur in human epilepsy? So here's our pathway that we discovered, our low-hanging fruit pathway in the bottom here. And if you take a section of epileptic tissue and you stain to see where these activated MAP kinase proteins, they're just on the surface of the brain. And we did this over and over again, and every time we do some of these things, it's all brand new, and we have to do lots of controls to convince us it's not an artifact. If you zoom in on this, this activation of this pathway is in these little arbors between the axons and dendrites, all this connected area, and also it spreads to the nucleus to signal to the nucleus. If you go downstream, those little pale areas in the middle of this stained area here, those are the cell bodies, and CREB we know is a nuclear transcription factor, and if MAP kinase is turning on CREB, you might expect it to be in layer 2-3 neurons, and that's exactly what we found. So this narrow band of activation here you can see pinpoints the area that we think some of this epileptic activity is coming from, because the genes were correlated to the activity, the pathways, we send localize where they are. And we start learning additionally interesting things. You can see on this high-power view here of the CREB and the nucleus that when you go into these recesses in the brain, it's all called the sulci, these pathways shut off. So the sulci in the brain seems to be acting as a natural barrier for the spread of these signaling pathways and probably these activities as well. The entire other cascade also, when you do in situ hybridization, localizes to that same level. So then you start asking questions like why layer 2-3 neurons? So we found that a lot of this activity correlated best with the spiking rather than the seizures. We know a little bit about cortical anatomy, that signals from the thalamus come up through layer 4, signals exit from the cortex through layer 5 and 6, but what layers 2 and 3 may do is form lateral connections. When this part of the brain wants to talk over here, they form connections in 2 and 3, and what are we dealing with in epilepsy? Synchrony. So how do you get large sheets hyperconnected and producing these synchronous discharges? It's through layer 2 and 3. So this is how we then come back and try to understand how you got there. But how did it get there in the first place? Why are these areas epileptic? One of the challenges we have is this. And I'm going to tell you about another method that we've come up with to try to characterize the tissue using these genomic measures called the cellular interactome. So as I mentioned at the beginning, many lesions, whether they're developmental or acquired, can cause epilepsy. Often the seizures, though, don't come from the lesion itself. If you have a brain tumor, it's the area next to it, the more normally structured neocortex. So then why is this? And if you look under the microscope, and we send it to our neuropathologist, and we have an outstanding one in the audience today, you get this report back. Oh, well, there's a little bit of gliosis, subcortical gliosis, and the white matter, maybe a little on the surface of the brain, but otherwise it looks like normal tissue most of the time. And depending on which pathologist you send it to, they'll tell you whether they think it's dysplasia or not. But bottom line is, this is not, there's no obvious thing of why this part of the brain is epileptic and another part is not. So can we use this information from these high throughput methods to kind of make sense of this? So one of my postdocs, Fabien Daché, did a kind of cool set of computational or mathematical experiments on these 1,700 genes. He just looked at their pattern. So these are 30 different human brain samples with different levels of epileptic spiking shown here. And this is a specific gene looking at the level of mRNA transcripts for a specific gene, say gene 1. And you can see on the right that there's high levels of transcripts, and on the left there's low levels. If you pick another gene randomly and look at its pattern of expression, these genes have nothing to do with each other. They're sort of inverted. The same brain samples don't have the same level of upregulation or downregulation of these two genes. Now, if you look across the 43,000 genes, you find incredible clusters of genes that kind of go up and down together. It's like a school bus full of kids. Sometimes the bus goes to the zoo. Sometimes it goes to the park or museum. If you could actually figure out who those kids are, and you could trace them back to what school they came from. So that's what Fabien did. And he hypothesized that each of these clusters was actually a different cell population that in the epileptic brain region was somehow different. Either there were different numbers of those cells, or they're phenotypically or genomically changed. So, but they're still very, very similar to each other to produce those clusters. So how did he do that? Together with actually an undergraduate in the lab, he wrote a kind of fun script that went to PubMed and took the list of genes in a given cluster, shown on this side here, took names of different cell types in the brain, and just did a reiterative PubMed search over and over and say, how many hits do you get? It's like when you do a PubMed search and you want to see how many hits you get for a certain number of things. And he took those numbers of hits and plotted them and basically suggested then that this group of genes that's upregulated in epileptic brain are endothelial cells, which means you might, if you stain that brain, see more blood vessel or a higher density of blood vessels in epileptic versus non-epileptic brain. So that's how it works. If you take the entire 1,700 genes, about half fall into clusters. They actually fall into 11 clusters. So we made predictions of 11 different cell type differences in epileptic versus non-epileptic tissue. Some of these, you can see here's the endothelial cell. This is a program called Cytoscape. So each of these dots is a different gene, and the closer the dots are, the closer they travel together in that school bus. So you can see how tight that is for the endothelial cells. Nearby the endothelial cell, we have red cells and white cells, as you would expect, because if you have more blood vessels, you're probably going to have more blood in that tissue. There's multiple populations of neurons, and there's some downregulated cell populations, an oligodendrocyte and a neuron type, and two inflammatory cell populations called microglia. So these all have very, very tight 95% correlation. You can then take the average of each of these and say, how do the cells change relative to each other? So mechanistically, are cells changing in response to other cells, and start modeling what's going on in the epileptic brain. And so this dendrogram shows how cells change relative to each other. And again, I think you can see this tight correlation between the blood cells and the endothelial cells. But then you see very interesting correlations, almost inverse correlations, between some cell types and others, that when one goes up, the other goes down. So you can actually start making predictions. So here's another dendrogram looking at changes as a function of spiking. And there's two green lines. These correspond to two different microglia or inflammatory cell populations in the brain. The shapes of the green lines are different, so we think they're doing different things. But they both go up. So you can say this is all kind of fun and nice, and it's math, and it's fun to make models and predictions, but is any of this true? We go back to the tissue, because we have the exact tissue. We stain with an antibody for microglia, in this case CD68. The high spiking area shows lots of them. The low spiking shows very few microglia. We predict more blood vessels. We actually stain the tissue. We look at the high spiking area. We see more tortuous blood vessels in the high spiking than low spiking. So everything basically that's been mathematically predicted using this approach, we've actually been able to discover. This is an interesting group here of populations of neurons and glia that are changing in an inverse pattern. This population of 280 genes is a neuronal population. It's downregulated. One of the genes we know is a gene called RORB, which is a layer 4 specific neuron. Another gene in this cluster is called NU-N, which is a nonspecific neuronal marker, a nuclear protein. So we think that if we stain the tissue that's epileptic, we're going to see a downregulation of these neurons in layer 4. The other thing we would predict from this cluster is that wherever these neurons go down, we're going to see microglia. So this is, again, looking at different tissue samples. Whenever this neuron population goes down, it's almost like you flip these curves over. But this is actually independent, 280 genes averaged together, and another 200 genes averaged together, showing this amazing reciprocal relationship between these two cell populations. So what did we find? It's something we call microlesions. So this is a new end stain of an epileptic human brain area. This is actually upside down, layers 1, 2, 3, 4, 5, 6. This is fairly normal. All the neurons are there. This area, which is kind of face to face, 1, 2, 3, 4, 5, 6, there's a big hole in layer 4, just as we predicted. And we showed this to our neuropathologist, and he said, look, the neurons are gone. And he said, no, they're not. And he said, yeah, look, they're gone. Well, he was looking at the H&E, the regular histological stain. The neurons are there. They look perfectly normal. But the new end stain of those neurons is gone. And if you look at a light microscopy level, the neurons are still there. Just the stain is gone. So it turns out to be a phenomenal tissue biomarker for these epileptic areas. And what we've learned about these microlesions is that they're messed up in terms of their synaptic connectivity. This is a stain for a protein called MAP2, which is a marker of all of the dendrites, the connections, and it's completely void. And as predicted, these areas are full of cells called microglia. And there's a very exciting data now from a number of folks suggesting that microglia are actually going in there and actually pruning or removing a lot of these synapses in these areas, but they're very, very focal. When we go back to many, many more tissue samples now, we see there's 3 different versions or types of microglia, microlesions in different layers, and the more microlesions you have, the more spiking you have clinically in those areas. So it goes back to the clinical for validating that these histological new lesions we discovered are actually real. So to summarize this part, we've discovered that the superficial layers of the brain, based on this mapping of these pathways of spiking, are in layers 2 and 3, and create perhaps this syncytium of hyperconnectivity, produce the spikes, and that this may result from these deeper layer of loss of synaptic connectivity, creating these new networks in the brain. Question, can we turn this into something useful? I mean, it's interesting observations. Can we make this into a therapeutic or something we can use to help our patients so we don't have to take out chunks of their brain anymore? And we're working on that, and I don't have time to go into it all today. Currently, a lot of the thinking about epilepsy is the brain gets injured, you have an insult to the brain, you have changes in the cellular and molecular responses to that, changing the electrical networks, creating the synchrony, producing the seizures. And it can take 6 months to years after a head injury or a stroke before people actually have their first seizures. And current drugs that we have now for epilepsy are all pretty much what we call anticonvulsants. If you take your pill, you won't have a seizure. If you have a seizure, it'll stop a seizure, but it does nothing to stop the underlying process. And if you miss a pill, that's the best way to induce a seizure in a patient with epilepsy. So there's a number of possible targets that come. We talked about the MAP kinase pathways. We talked about inflammation and microglia and neovascularization. So we can even repurpose existing drugs now that we have clear pathways to go after. So clearly the next step and something we're doing is animal studies to validate that these targets truly are cause and effect of epilepsy, and we can develop drugs against them. But to get to the clinical trials, I wanted to talk about another approach that we've taken, another aspect, because what we really need for clinical trials are clinical biomarkers, surrogate biomarkers. Can we do an MRI and see if somebody's going to get epilepsy, or do some kind of test, non-invasively or a blood test, and say, you're at risk for epilepsy, let's take this patient group and let's treat them in our drugs that we're developing to prevent epilepsy. So this is the problem we have with our gold standard. We talked about the electroencephalogram, the EEG, and this shows a comparison of what you can see on the scalp on the right-hand side. You can see these little sharp waves here and here, 1 and 2, but everything between just looks like a bunch of squiggly lines. And if you compare that on the same patient to intracranial electrodes, you can not only see those same spikes, but look at all the stuff that's going on in between it. So really what we need to get to as a level of resolution of this in a non-invasive way. If we have to take everybody's skull off and put electrodes, that's probably not a good way to screen large populations to see if they're going to get epilepsy. So we started focusing on another aspect of our systems biology, and instead of looking at RNA, we looked at metabolites. Because metabolites are something you can actually measure non-invasively through MRI technology, through magnets. So this is the metabolomic profile of a piece of epileptic tissue from the same area that we see induce these genes and RNA features. And you can see very, very nice resolution of those areas. And we can do the same experiment. We can take areas that have epileptic activity, areas that don't, put it in this 12 Tesla magnet, and try to see if we can find a uniform signature of metabolites that's predictive and sensitive and specific for epileptic brain areas. that might someday help us non-invasively see these things. And that's what we have done, at least at the tissue level. This is ex vivo data. And you can see a very nice separation using this logistic regression model. If you take a series of metabolites and they have this pattern, this goes down, this goes up, this goes down, it's like a little magic formula, like a combination lock that says that's an epileptic brain area. And it has very nice sensitivity and specificity. So the next step is how to turn this non-invasive method, invasive method, can't take tissue to do this study, into non-invasive. And we're working with folks here and others to really turn this into a 3D non-invasive model to see epileptic activity that we can sometimes use between human and rat studies to diagnose and treat patients with epilepsy, even long before they actually show their first seizure. So I want to kind of finish up with how do we put this all together? I gave you a couple of examples of how we probed for layer-specific differences, understanding the mechanism of epilepsy, finding drug targets, and developing biomarkers. But I think the potential of this systems biology approach is enormous to look at so many different things that you can link together. Any part in somebody in this room, I'm interested in biochemistry, I'm interested in gene editing, I'm interested in electrophysiology or brain structure or imaging of the brain, can be linked to all these other variables. So let me give you an example of how we can start to tie all this together. And it's very simple. If you find two things in your database, in this case, a neuron type 4 in yellow and a nuclear protein in blue, you can see that they have a correlation, a positive correlation to each other. So you can create a linkage in your database. Here's an example of that same neuron population with a long non-coding RNA that has a reciprocal. And again, you define what's the cutoff statistically. So we use 0.9, 0.95, something like that. So these have a negative correlation. And you can take all of the data in the database on all these different molecular, clinical, imaging, other types of variables, and build a matrix. And when you put it together and you plot it back in this program again, it looks something like this. Anybody who comes to Chicago in the summer on Wednesday and Saturday nights and look over Navy Pier, this is what you see. It's fireworks. But basically, each of these symbols represents a different type of molecule. You probably can't see this, but tiny little circles are either coding or long non-coding RNAs. We have metabolites as little red squares. Different types of proteins, membrane, nuclear and cytoplasmic proteins, different cell types as the yellow circles, medications, age, EEG characteristics. And you can see what relates to what with completely unbiased discovery tools. So let's take this cluster here. This is actually a red blood cell cluster. So when you look at the proteomics and you put that together with the cells, not surprisingly, certain blood proteins like hemoglobin, fibrinogens, IgG molecules enter that cluster. Of course, why wouldn't they? But what's cool is all the things that you don't expect to be in that cluster. What are these metabolites that correlate with more blood in the brain? What are these clinical variables? Oh, what's this? That's a medication. And you see that's a medication called Vimpat, and it's used in patients with epilepsy. Why does this come in this cluster? I don't know, but the company that makes it might be interested in knowing maybe why they have some anemia in their patients who have epilepsy. Here's another example. These are our age-dependent variables. So we have patient samples from the ages of 6 months to 50 years. Are there certain genes, proteins, molecules, metabolites that are different in babies versus middle-aged kids versus adults and so forth? So we can select for age-dependent variables, and we get groups of genes, proteins, molecules, and we get another medication, called vigabatrin. The reason that comes in this cluster is because it's something that's only given to babies, actually for infantile spasms. So it kind of then makes sense, and as we build this database, it allows us to find a lot of new things that we never thought of before, and really is amazing discovery. So to summarize, this is the idea of total data integration, combining high-resolution, personalized data sets together, identified both patient-specific, but really, if we're going to think of developing drugs, the common ones are our low-hanging fruit. Those are the ones that are going to help the most patients the quickest. So what are the next steps? So since moving here and building and working with our CCTS and biorepository, and can we bring all this together? And so we've created this sort of big data dream, pretty much doing what we've done before, but at a much higher level, taking the entire electronic health record of a hospital system, which exists on one server, taking an imaging server, completely de-identifying it, but linking the patient records to both. Histology, so if we have tissue, first of all, we need to know where it is. And that's really critical, having a pinpoint on your imaging, a little X there, that we can say that's where this tissue comes from. And so if you are going to then look at your histology, it's going to be linked to the imaging. It's going to be linked to any omics, any of the studies you do. It's going to be linked to any electrical signals in the case of epilepsy patients, so you'll know what's going on there. You want to link to external servers, tissue inventory, and then really come up with creative data mining tools to find interesting cures to diseases as well as biomarkers. So I'm going to stop there. This kind of work takes a lot of help from a lot of collaborators from all around, and thank you for your attention.